prokaryotes enveloped and its modification. Okay, student. First, we discuss envelope of prokaryotic cell. And prokaryotic envelope mainly 3 layers. Okay. You know one of these, cell wall, then another is plasma membrane, and outermost layer is glycocalyx. Okay, student. Prokaryotic envelope. Prokaryotic envelope is made up of mainly 3 layers: plasma membrane, cell wall, and glycocalyx. Okay, student. In which first we discuss this glycocalyx, which is outermost layer. This glycocalyx, if present as a loosely, if glycocalyx layer is present loosely, that is known as a slime layer. That is known as a slime layer. If glycocalyx present in a form of thick and tightly bound, so that layer is known as a capsule. Okay, student. Outermost layer of the prokaryote cell envelope made up of glycocalyx. This glycocalyx, if this glycocalyx layer is loosely attached, that known as a slime layer. And if this glycocalyx layer is thick, tightly, that is known as a capsule. Okay. Further, next layer is cell wall. Student, cell wall of this prokaryotic cell made up of which substance? You know? Yes, peptidoglycan. We discussed in the last lecture, right? Here, this cell wall give proper shape of this cell. If in cell wall cell wall made up of peptidoglycan. If peptidoglycan layer is thin and if thick, student, here there are 2 types of bacteria. Some of the bacteria have a thick cell wall of peptidoglycan and some of the bacteria have a thin wall of peptidoglycan. Here, one of the important thing is Graham's scientists use this Graham staining method. Basis of this Graham staining method, the cell wall of bacteria may be represented as a negative or positive. How? When we apply this Graham stain into prokaryote bacteria, thick cell wall of the peptidoglycan absorb maximum stain. So they absorb as a dark violet color. Whereas if bacteria have a thin cell wall, so they does not capture any more stain, so that is pink in color. So that type of group of organisms known as the gram-negative. So basis of this gram staining, cell wall of bacteria or cell envelope of bacteria divide into two types, gram-negative and gram-positive. And why this negative and positive? Because of their presence of cell wall. If cell wall is thick, they are negative. If thick, they are positive. Okay, students, with the which help of the cell envelope divided, then it is gram staining method. Okay. Now, here to separate these things. Okay. Next one is peptidoglycan. Okay. Now, plasma membrane. Modifications of plasma membrane extend into form of tubules, lamellae. Okay. This extension of plasma membrane form tubules and lamella-like structure. These modifications of tubules or lamella-like structure modification of tubules and lamella-like structure known as this one is known as we discussed in the last lecture, mesosomes, right? So this modification tubule and lamella-like structure known as mesosomes. Okay, student, these mesosomes are extend from this plasma membrane enfolding-like structure. These mesosomes have a function like respiration, then replication, then cell wall formation, then increase surface area of the cell, then increase enzymes into plasma membrane. Okay, student, mesosomes is modifications part or modified of this plasma membrane. Okay, this form tubules and lamella-like structures. These mesosomes have a respiration process. Respiration is here in that for used energy. Okay, we use how here respiration is formed like mitochondria. How mitochondria use glucose and form energy and a form of ATP. Here also respiration act as a form energy. Okay, so in bacteria or in prokaryote, mesosomes act like a mitochondria. Remember, student, okay, form of energy. Then replications means here this one is DNA. If DNA separated and two new form of DNA form, this is known as a replications of DNA. Okay, एक DNA में से नए दो DNA bond बनने की process, that is known as the DNA replications. Then cell wall formations. So cell wall formations में भी ये mesosomes helpful है. Then increase surface area. जो bacterial cell है, इसकी जो surface area है, उसे increase करने में help करता है. Then also these mesosomes maintain enzymes into the plasma membrane area. Okay. Then another form of extension of plasma membrane. Important thing is extension of plasma membrane known as a chromatophore act as a pigment. Okay. Extension of plasma membrane. Here some of the extension of plasma membrane. This is formed from the plasma membrane. This extension of plasma membrane known as a chromatophore, which is act like a pigment. Okay. Here this is also modifications part of the plasma membrane. Okay, student. So here we discuss modifications of envelope. Okay, in which we discuss outermost layer, glycocalyx. It's have a two type, slimy layer and capsule. Then we discuss cell wall. Cell wall it's have a thin or thick or gram positive or gram negative. Then last one we discuss plasma membrane modification known as a mesosomes and their functions. Okay, किस तरह से ये mesosomes function करता है. तो रेस्पिरेशंस में यानी कि एनर्जी फॉर्मेशंस में, रेप्लिकेशंस में यानी कि डीएनए मल्टीप्लिकेशंस में, सेल वॉल फॉर्मेशंस में, बैक्टीरिया सेल की सरफेस एरिया इंक्रीज करता है, देन जो एंजाइम्स है उसे मेंटेन करता है. उसके अलावा सेल प्लाज्मा मेम्ब्रेन में से एक्सटेंशन ऑफ प्लाज्मा मेम्ब्रेन नोन एज अ क्रोमेटोफोर, प्लाज्मा मेम्ब्रेन में से एक्सटेंड होकर क्रोमेटोफोर्स लाइक स्ट्रक्चर्स बनाता है. दैट इज आल्सो one kind of pigment. Okay, student. So here these things, prokaryote envelope and modifications. Thank you.